Hello, my name is Sergey Malkovich. I've been a American scientist since I was exiled from Russia for experimenting with gene editing. My sole goal is to push the human's limits and maximize our human capabilities. I will be meeting with subject 626 in the morning. Good morning, it is I, Sergey Malkovich. My doctor in training, Dr. McDreamy, will conduct the interview with subject 626. I will provide any further explanation if needed.
Hi, I'm Dr. McDreamy. How will you be altering my baby? Please watch this video.
What is the role of CRISPR in bacterial immunity? First, we must define what CRISPR is. CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. Now, in the scientific community, we first started noticing CRISPR in bacterial DNA. So let's take a look at that. We have our DNA strand going along here. And what we noticed is that in bacterial DNA, there are your normal bacteria DNA, and then there's also these things called spacer DNA. And these spacer DNAs are identical to viral DNAs that are harmful to the cell. Now, take a look over here. We have what's called Cas genes. Take a look at that. We'll talk more about that in a second. Let's take a better look at this spacer DNA by blowing it up. Here you have your spacer DNA. You have a Cas RNA complex, and this will insert itself into the spacer DNA, transcribe itself, and form a structure called CRISPR RNA. Here you have your tracer DNA as well. Right off the end, it forms a hairpin structure. Now let's take a look at what the role of CRISPR RNA is. Here you have a Cas protein, very complex structure, and that comes from the Cas genes. It is transcribed and translated to form this very unique structure. And the CRISPR RNA will insert itself and form bonds with the Cas protein. And what this will do ultimately is when the viral DNA is injected into the cell, this will hunt that DNA and terminate it, killing it, rendering it useless. Now, I know what you might be thinking. What happens if a new virus comes in and injects its DNA into the cell? What's cool about this part is that Cas protein will actually take a bit of the viral DNA, the new viral DNA, take a little copy of it, and insert it into the CRISPR DNA found up there. So you'll have a portion of DNA that is now that new viral DNA, and that helps with the bacterial immunity. So, how do we ask about the big picture of CRISPR and bacterial immunity? Well, CRISPR is a bacteria's immunity system.
CRISPR-Cas9 in gene editing is a new technology that scientists are really excited about because it's cheaper, more accurate, and more effective than other methods of gene editing. So, it works by using an RNA sequence with a guiding sequence, a small guiding sequence that can attach to specific parts of a DNA sequence. And it also attaches to the Cas9 enzyme, which will actually split the sequence at the particular point where the guide sequence attaches. So that brings new ways to edit DNA. So one way to edit DNA is to add. So all scientists have to do is splice the DNA and let the DNA repair machinery repair where it's broken. This will add genetic material. The second method is deletion. Scientists have to cut the DNA in two spots, and then they can actually remove this fragment, and the DNA repair machinery will repair these two parts together. The third way is to replace. Replacement is similar to deletion, where scientists splice the cell in two places to create a section for removal. So they will remove this section and add a custom sequence of genes into that section. The big picture, this is how CRISPR-Cas9 can be used in gene editing.
In regards to Cas9 and CRISPR, we are at limitations currently, and there are ethical dilemmas revolving around it. Currently, we're only allowed to do single generation changes due to the fact that many countries have banned the gene editing of gametes, meaning the sex cells are off limits. We're not allowed to edit it. Not allowed to edit. And perhaps that's for good reason or bad. That's up to you to decide. In terms of bioethics, we have to be careful about future class divides, potential social divides, and maybe even the potential of advancing humanity together as a whole and creating a higher functioning society. It all depends on us and how we deal with it. So it's important for us to maintain and think about how CRISPR and Cas9 affects us and how it will affect us. We even have the potential to grow our own organs and give ourselves transplants of the same identical organ. Meaning if I have a bad heart and I grow a brand new good one that has my DNA coding, I could replace my heart and it'd be perfectly well, perfectly healthy, without the repercussions and consequences of having a different heart transplant from another person. After watching our current video, do you have any questions?
What will my baby look like?
Baby will look like this.